All right.
Well, welcome everybody. My name is Tommy Soares. I'm Assistant Director of the Purdue Institute of Inflammation, Immunology, and Infectious Disease. And I want to welcome you to another Purdue Lecture Hall series where today we're honored to welcome Olivia Robertson from the Department of Human Development and Family Studies. Olivia is doing some really interesting work on looking at stress during pregnancy and looking at hormone levels in hair. And this is awesome work. I'm so interested to hear how you got into this and how's it going and well, everything and all about it. So without further ado, please welcome Olivia Robertson. Thank you for speaking with us today.
Yeah, thank you so much. So hi, everyone. Thank you all for tuning in to this talk. Really excited to be a part of the lecture hall series and get a chance to talk to you all, tell you a bit about how I got into research as an undergrad and now as a graduate student. And so my name is Olivia Robertson, and I'm a fourth year PhD student in the Department of Human Development and Family Studies. So I'm going to start a bit by just telling you a bit about myself, how I got into research, and then I'll tell you a little bit about a project that my lab and I did here at Purdue in the West-Lafayette area, which was a pilot study that looked at the correspondence between objective measures of stress from hair cortisol and subjective measures of stress, for example, like self-reported anxiety. Okay, so actually, first of all, I'm originally from Canada, and specifically, I'm from the capital city, Ottawa, and I grew up there all my life until I was 19, and then I left home to go to school in the US. And then so I ended up going to school in Iowa, of all places, and specifically, I went to Iowa State University, which is in Ames, Iowa, which is a small college town similar to West-Lafayette. And the big reason I went to Iowa State is because I was recruited to run cross country and track and field there on an athletic scholarship. And so this is me as a freshman in my glory days running a relay at the Big 12 indoor track meet. But when I was in high school, I wasn't really sure what I wanted to do for a career, but I knew I was interested in sports and endurance training and a little bit of biology, but I still wasn't 100% sure about what I wanted to do. But when I started school, I enrolled as a kinesiology major, specifically in the exercise science stream for my first semester. But then in my fall semester, I was more and more unsure of what I wanted to do, and I was exploring other majors. And then by the spring semester of my freshman year, I thought maybe I wanted to do some sports media type work or work in a collegiate athletic department. And so then I did that for like a semester or two in a summer. But then the more classes I took, I realized that I wasn't as interested in public relations as I thought I was. But what I did really like was the theoretical piece and the theoretical courses I took on public relations, which turns out to actually be a lot of applied psychology. So then I realized that really is what I was interested in was psychology. So then that leads me to how I got into research as an undergrad around the end of my sophomore year or so. So after I got into psychology, I really dove into working in all sorts of research labs, anywhere that would take me. At first, I volunteered just at the end of a semester just to get any experience, and I did a little bit over the summer too. And then in my junior and senior years, I got involved in a few different labs a little more seriously. One of those was the Network Community Counseling Services, which is both a research lab and also a clinical practice setting for delivering therapy to both students at Iowa State and also community members. So this lab is run by a counseling psychology professor at Iowa State, and it's also where the graduate students who are learning how to become counselors get to practice and train. So as an undergrad research assistant, we were in charge of setting up the therapy room, taking payments from the clients, and helping with any other kinds of administrative work to keep the clinic running smoothly. And then another lab I worked in was the identity development lab. And as a research assistant in that lab, I mostly helped collect data from undergrad research participants on the topics of stereotypes of different identities, So for example, student athletes and LGBTQ identities. And then here is a picture of me and one of my mentors, Fred Clavel, for a presentation I did at an undergrad research symposium as a senior, which was basically my first kind of go around at coming up with a research question, doing some statistical analyses, reporting my findings, and then discussing them in a 15-minute conference style presentation to other undergrads and mentors at the symposium. And Purdue also has these every semester, and they're a great way to learn about the research process and to learn how to communicate science as you're still kind of learning and getting the hang of things. So then finally, the lab that I spent the most time and is the most similar to the research I do now was the SPIT lab led by Dr. Bertie Shirtcliffe. And the SPIT lab is a stress physiology lab that mostly focuses on understanding how experiences, particularly stressful experiences, influence our biology, or the kind of popular expression is how stress gets under the skin. So how do stressful experiences biologically embed? And so the SPIT lab is a, oh, sorry, that's me in the picture. And then so as the name suggests, the SPIT lab does indeed use spit or saliva to understand stress physiology. So in particular, working this lab, I learned how to conduct analyses on hormones found in saliva. So for example, cortisol, which is typically considered to be the main stress hormone, but also for testosterone, DHEA, and estradiol. And then I also learned how to do those analyses for hormones coming from hair, and that's mostly what I'm going to focus on and share about today. And then the picture, this is me spitting into a tube, which is how we literally collect some of the data for saliva. And then in this picture, this is me getting my hair cut for some videos that we did to teach other researchers how to collect hair. Okay, so then I graduated, but before that, I knew that I really enjoyed research, but again, I was very undecided of what I wanted to study. But with the help of my mentor, Bertie Shirtcliffe, I ended up applying to graduate school at Purdue to work with some of her collaborators of hers who were open to me about not being totally sure about what I wanted to study, which was, that was a big stressor for me in terms of wanting to decide to go to graduate school. So thankfully, it all kind of worked out, and I think I have a good fit in my lab. And as I've gone on, I've been able to narrow down what my research interest is in, and I've so far really enjoyed my time in graduate school. And then so here, this is my lab. So on the left here is my advisor, and she is the director, Dr. Christine Marceau, and she's the director of the Biobehavioral Development Lab. And so our lab is generally focused on understanding genetic and environmental influences, as well as prenatal or before birth influences for various child and adolescent outcomes. And so my advisor's research is currently focused on understanding things like adolescent substance use, but we all study lots of different outcomes such as childhood obesity, child behavior problems, bullying, and a lot of other topics. And then so the other individuals pictured here are my wonderful lab mates, Amy, Hazel, Nicolina, and Sohi, and they're all younger than me in the program, so I'm kind of like their big sister. Hopefully I'm a good role model for them. So the specific project that I want to tell you all about today is the pilot study that my lab did a couple of years back, which is called the Maternal Context of Pregnancy Project, which is a study of pregnant women and their experiences during pregnancy. And this study also included a non-pregnant comparison group, so we could compare what we found in pregnant women to non-pregnant women as a sort of a baseline. So here is kind of like the overall objective of the MCPP, and that was to understand how different types of stress relate to each other. So for instance, cortisol as measured by hair hormone concentration and how that relates to self-reported symptoms of, for example, depression or anxiety. We also wanted to note how those different types of stress relate to birth and infant outcomes, and then also whether the stress experienced during pregnancy and the resulting birth and infant outcomes are mediated by epigenetic changes in the placenta. So these are the specific goals of the project, and I'm specifically going to talk to this first one, which was to better understand how objective measures of stress, such as hair cortisol, relate to subjective measures of stress, like anxiety or depression. And so we hypothesized that the higher the pregnant women's hair cortisol was, the more psychological distress or subjective stress that they would report. So MCPP is a study of 34 pregnant women and 34 non-pregnant women from the West-Lafayette area that we followed over time. So basically this graphic you're looking at is a summary of the study timeline. So we first recruited pregnant women before the end of their first trimester and had them answer surveys and questionnaires about themselves and their levels of stress. And we also cut their hair at that time too. And then we did that at the end of each trimester. So trimester 1, 2, and 3. And then at the time of birth, Emily Rowland, who is a former graduate student in the lab, who's now a postdoctoral researcher at Michigan State University, went to each hospital at the time of the birth at all hours of the night and collected the placenta that were used in the epigenetic arm of the project. And then below we have the kind of complementary side of the study for the non-pregnant women, which we followed on a similar time, on this exact same timeline, just they weren't pregnant. So we just had visits with them every 3 months. And importantly, we collected hair at the end of their trimesters because hair is a retrospective measure of stress exposure. And we're going to talk about that just next. So about this hair business and what it can what it can tell us about stress. So previous research has found that on average, hair grows about 1 centimeter per month or so, and it kind of slowly seeps into the hair follicle. So as you can hopefully see in this figure on the left, cortisol, these little dots, is leaving the bloodstream and entering the hair follicle. And then on the right to it, you can see this ruler that's showing like 1 centimeter from the scalp, 2 centimeters from the scalp. That's kind of a loose measure of the time of exposure for that length of hair. So briefly, what we believe is happening is that the body is having a stress response, which is governed by the hypothalamic pituitary adrenal axis, or the HPA axis for short. So for example, when something stresses you out, like for example, an upcoming exam, and you feel stressed, then the middle structure here, the hypothalamus, will secrete corticotrophin releasing hormone, or CRH, and that tells your pituitary gland to produce adrenocorticotropic hormone, or ACTH. And then that ACTH gets transported to your adrenal glands, which they sit on top of your kidneys, kind of in your mid-back area. And the adrenal gland is what secretes cortisol. So what we think is happening is that the more stress responses somebody is having, the more and more cortisol is seeping into their hair. So that's why we think that people who are more stressed are going to have higher hair cortisol. And then, so now I'm going to show you a video of a hair collection, which is essentially what Emily and I did for all the participants in the MCPP study that we collected their hair.
6 straight youth hair, straight part. This video demonstrates how to collect a hair sample from youth or adults with 6 straight hair using the straight part method. Supplies for hair collection include a large hair tie, small rubber bands, hair clips, and a pull sheet. We recommend using sharp scissors with a fine point capable of cutting close to the scalp. Part the hair at the back of the head. The part should be perpendicular to the center of the ear, and the sample will come from the bottom third portion of the scalp. After the initial part, use a large hair tie to secure the hair atop the head. It is okay if the participant completes this step themselves. This step isolates only the hair that will be collected. Use the clips to keep stray hairs away and ensure that only the intended area of hair will be snipped by the scissors. Use the comb to make a straight part below the initial part. This is the hair that will be used for the hair sample. Here, the sample collector has sectioned off a large sample. The straight part method does not require a huge hair sample, so the sample collector repeats the part to avoid participant embarrassment. The straight part is ideal for 4 biomarkers, but might be too large for youth with thinner hair. The sample collector may opt to take 2 to 3 smaller parts. The video demonstrates a part in the middle of the scalp, but we recommend collecting towards one side so the sample is invisible with all the hairstyles, including pigtails or braids. Use a small rubber band to tie off only the parted segments of hair. The rubber band should be close to the scalp. The rubber band tells technicians which end of the hair sample is the scalp end. Next, gently pull the sample taut before carefully snipping away the segmented hair strands. It is important to get as close to the scalp as possible while being careful not to nip the participant. Work slowly and carefully to provide a neat and tidy sample. This is enough hair for most projects to assay 4 biomarkers, with enough remaining sample to check for accuracy. If the straight part attained less hair, simply repeat the process until 100 to 150 strands are obtained. After completing the collection, the participant's hair is lightly fluffed. There are no visibly noticeable sections of missing hair. Hair is stored in a foil sheet. Check out the sample packaging video for shipping instructions. The lab technician has isolated the 3 centimeters of hair that are closest to the scalp and weighs the sample precisely. The sample collector obtained 163.9 milligrams of hair, a very good hair sample, without leaving any noticeably missing hair on the participant.
Okay,
Big
so
straight youth.
that's what a hair collection typically looks like. Those were instructional videos that my old lab created to help other researchers collect hair, and so that gives you some insight into what that's like. So after we cut the hair, so now I'm going to tell you about after we cut the hair ourselves, we packaged it off and actually sent it back to Iowa State University, where the spit lab analyzed the hair for hormones, and then they sent us the results. And now I'm going to walk through how I used to do that when I was undergrad working in the spit lab. So first, hair is cut to the desired length from the scalp end. So in our project, that was 3 centimeters worth because we wanted to capture the last 3 months exposure of cortisol. So these are examples of some hair sections that I've cut before. And so we make sure to mark in this little sticker, you can see it says cut end because we want to make sure that we have the right end, which is coming from the scalp, the right side up. And we also wrap them in tin foil because any light exposure can sometimes damage the hair and reduce the amount of hormone there. So after we cut the hair to the desired length, we have to wash the hair. So we wash the hair to just strip it of any oils or dirt or other impurities. So the hair gets washed twice in isopropanol, which is a type of alcohol, for 3 minutes using an inverting rotator. So in this picture, we have these test tubes here that we put the hair in, and then we fill up the test tubes with a little bit of alcohol, and then they rotate for several minutes. And then we dump the alcohol out and put some more in, and then we repeat that so that it gets washed twice. After that, the washed hair is then dried under forced air for about 4 to 5 hours to ensure that all the alcohol has evaporated. And then once it's dry, we do the weighing. So exactly 15 milligrams is weighed out using a very precise scale. And the scale is so sensitive that if the door of the lab is open and there's like a breeze, the reading on it will be different. So it's very precise. And then so the hair is in this small test tube, and then after that we grind it up. So we grind it up into a powder, and we do that using this machine that's in the picture here. And so you can't see it, but in those white boxes, you can take the lid off, and then there's little slots that we can put those orange little tubes in. And then basically we close it up and clamp it down, and then this machine basically just shakes it back and forth. Oh, and we also put in 3 stainless steel beads, and that is kind of what breaks up the hair. And so it just goes so fast for about 5 minutes that then the hair is reduced just to basically a powder. You'll see that the picture is blurry because this is a picture of it in motion, so it was going really fast. And then after we grind the hair up, we do the extraction step. And for that, we add about 1.5 milliliters of methanol, which is a methyl alcohol and solvent that extracts the hormone from the hair powder. And so the tubes, as you can see in the picture, are placed on this rotator, and they rotate for 24 hours at room temperature. And that's basically how we get the hormone out of the hair. Once that's complete, we centrifuge the samples. So this picture is pretty zoomed in, but basically it's this basin where we put the little orange tubes in, as you can see in the picture, and then the lid comes down, and then it basically spins it really, really fast so that all the powder of the hair is down at the bottom. And then once that's ready, we take out 1 milliliter of the hair and methanol, and then we put them into new orange little tubes. And after that's done, we evaporate all the methanol out of the tube. So we dry it down under a stream of nitrogen gas in an evaporator at about 50 degrees Celsius for 20 to 30 minutes. So after that, there's no liquid in the tubes. There's just kind of like a slight like powder residue. And then once that's done, we add about precisely 400 microliters of a diluent to reconstitute or reform the hormone liquid. And then we vortex the tubes, and that's just what the machine is called. It just basically mixes up the diluent and what's left of the hormone, just to make sure that it's all uniform. And then once that's ready, the samples are ready to be analyzed for their hormone concentration. So specifically in this lab, we use an enzyme immunoassay for detecting and quantifying the level of hormone concentration for each participant sample. And so I don't think we have time to explain the whole process, but it takes anywhere from 3 to 6 hours, depending on what hormone we're analyzing for. And then just to compare to saliva samples, this is where we would start the saliva assay. So you can see in the picture, there's the orange tubes from the hair, but then there's this taller tubes, and that's just, that's a saliva in it. So this is kind of all the steps that we did before was just to get the hair into a liquid format so we could pipette it into these small wells and do the rest of the procedure to find out what the concentration of the hormone is. Okay, so what we found, so after we got the results back, we analyzed the data that we got. And so basically we did find what we were expecting, that having a higher hair cortisol concentration was associated with reporting more symptoms of overall subjective stress, symptoms of anxiety, and then pregnancy-specific anxieties, in pregnant women. But surprisingly, we didn't find this for all measures of stress. For example, we didn't find that for depression, but in general, we did find that higher hair cortisol was associated with higher subjective stress. And so what this study allows us to better understand is how the underlying stress physiology maps on to pregnant women's lived experience. And this will hopefully give us insight into better understanding how maternal stress influences child outcomes, which is what we're looking at doing next. So I've actually gotten the opportunity to present these, the preliminary version of these findings at an online conference about 2 summers ago, which was the annual meeting for the International Society of Psychoneuroendocrinology. And then just a couple weeks ago, I submitted a paper of our findings for this project to the Journal of Hormones and Behavior, and hopefully it will be published sometime so other scientists and people can read about what we found and what we did. Future directions for this project are to include investigating the effect of subjective and objective stress measures on the birth and infant outcomes themselves. And in fact, one of our undergrads in our lab did an oral presentation at one of the Purdue research symposiums last spring. And so Julia was interested in looking at negative life events experienced during pregnancy and seeing whether or not that related to infant temperament. And that was based on previous literature that suggests that experiencing more stressful life events for moms during a pregnancy is associated with more difficult or anxious temperaments in the infants. And so counter to the prior literature, she did not find an effect of negative life events, but she did find that lower hair cortisol was associated with more negative temperaments in the infants, which is a kind of counter to what we were expecting, but we're going to be looking into that more in the future, hopefully. And then another one of my lab mates is looking into more the epigenetic side of the project, and we're really excited about what those findings may bring. So that's all that I've got for you today, and I'm happy to take any questions, and I've included my email here in case you want to reach out afterwards.
Awesome. Awesome, Olivia. So interesting. Thank you so much for doing such a great job to tell us a little bit about your journey and how you got to where you are now. So exciting also to see you being able to publish your work and be able to present it at conferences. Tell us a little bit what that is like. Is that very stressful? I know we've been talking a lot about stress and stress hormones, but is it very stressful to do these publications, to present your work at conferences?
Yeah, it can be. I think that one in particular was less stressful because I got to just record my talk. So I could prepare a lot. So I didn't have to speak in front of a large group or anything, which was good. But other conferences, it can be stressful if they're in person and you have to talk in front of a big group of people. But generally, my experience has been positive, and it helps when you have kind of people to come support you and your advisors there, and just to make it feel more comfortable. Yeah, and I'm really excited about getting that paper submitted. And that takes a long time, but again, having like the support of my advisor, it was kind of like teaching me how to kind of do the research process and stuff. And I think now that I'm in my 4th year, I'm kind of getting the hang of it, and I've submitted a couple papers now. So it's definitely difficult, but there's definitely, hopefully you'll have a lot of support available to make it easier and help your learning. Yeah.
Absolutely. And, I think it's important also to be able to reflect on what work you've done. And so the presentation, putting the presentation together or putting that paper together helps you to look back and say, hey, I've done this, I've done that, I've done these experiments, this has led us down this path. And so that type of reflection is important also to feel proud of your accomplishments, because I would feel proud of your accomplishments. I'm very proud already of the work that you've done, and these videos, and such a diverse experience to research. A lot of the work that you do extracting the hormones from hair remind me of the work I used to do extracting metabolites in plants. And it's very much the same process of, extraction and drying down and making sure you're very accurate with your measurements. And, tell me something. A lot of the students do ask, does it take a lot of math to do research? And you were very precise about your measurements, and there's probably a reason why you had to be so precise, and maybe it's a mathematical reason too, right? So does it take a lot of math knowledge, you think?
I mean, specifically for those procedures, I mean, it's important to be precise because that's what they've been, validated for. I'm not sure if that's necessarily math, so much of it is just being precise and following the instructions. I guess in my field, generally, it's, I think the most math heavy thing we do is using a lot of statistics. I find in my field, though, since it's very applied, I think there's more emphasis on maybe a conceptual understanding of the stats, as opposed to kind of a heavy emphasis on the kind of underlying mathematics of things. I know like that was something I struggled with a bit with like math courses, but I find that the way that, I don't know, my statistics courses have gone here, kind of emphasize more applying the appropriate test. So it's similar but different. So I find, I mean, if that's a deterrent to people wanting to go into science, I think there's a lot of, I know a lot of people in my program and a lot of professors who are not like math whizzes, and they still do very good work. So I hope that won't stop people. Yeah.
Awesome. I agree. There's so many areas of research that for the heavy math whizzes, there's science for them to do, and for the ones that are not so heavy on the math, there's science for them to do. There's a lot of science to be done around. Olivia, you talked a little bit about how you participated in the research with the Community Counseling Services Network.
Yeah.
How did you get started? Like, did you go talk to somebody about it? Did you find some information? Do you remember how you got started or how you found that placement?
Yeah, so a couple of different ways, I guess. I'm sure it's similar here. I don't know if it's exactly the same, but at Iowa State, once I became like a psych major, or maybe even before that, I got onto like the psychology department's mailing list, and they would send out like research ads looking for undergrad research assistants, so labs that were looking for help. And sometimes I volunteered, but most of the time I took it for credit, which I know like almost all, Purdue does, and almost all the big universities do as well. So that was one avenue. So kind of just reaching out to kind of like a job ad kind of thing. But then also, actually, when I got involved into the SPIT lab was in, so I was a psychology major, but the SPIT lab was in the Department of Human Development and Family Studies, which is the department I'm in now at Purdue. And so I wasn't in that major, but I just kind of like looked up the faculty profiles, and because I was interested in biology and social science, and I saw like, oh, they do really cool things with hormones and saliva and hair. And so I honestly just emailed the faculty member, Bertie Shercliffe, and she was like, yeah, come to a lab meeting. And I just came to a lab meeting, and then I was in the lab, and and then I learned like so much from them. So I think just reaching out, if there's not like a formal way, just reach out over email or however. And I think oftentimes professors are always looking to engage with students. So yeah.
That is so important. I resonate with that piece of advice, because it takes people kind of getting out of their comfort zone and going to talk to people, right? And asking questions and being curious. And, this whole thing of research doesn't just come quickly or naturally. You have to kind of explore it, right? And explore what type of research you like to do. That SPIT lab experience must have been awesome. I can't imagine. I want to ask you about like how do you manipulate spit and so on, and saliva, and like all the components and like preservatives and things, whatever. But that must have been an awesome experience. So is this spit lab, was this a program that you were doing that was funded by a government agency or a company? Do you recall how that was happening?
Yeah, so there was a couple different things going on in the lab at the time. So yeah, the faculty member there, Brady Cliff, she had her own projects going that I worked on, but she also would consult for people's projects. So for example, I would work on studies that I think they were studies of adolescence. So kids going through puberty, and there's lots of hormone changes going on at that time. So I think that was out of the University of California, Berkeley. And so they would send the samples to the spit lab, and then we would analyze them for it. And that was part of, I believe, like a NIH National Institute of Health grant. Other times we were working on like smaller studies. A couple of the grad students had like pilot studies or like dissertation studies kind of thing that we would analyze samples for. Yeah, and then a couple projects in the lab were just kind of validation studies for different hormones and stuff. So there was a lot of different projects going on, and some of them were government funded or, and then I think one other project that is a little bit of an industry partnership, I think, is to get at like an instantaneous reading of cortisol. So that's kind of like a research and development product. Yeah, because obviously the analyses for saliva and hair, they take lots of days and hours, and so they're working on like a prototype for kind of getting like an instantaneous reading of that. So that was one other thing. Yeah.
Awesome. That would be so neat. A fast cortisol reader
Yeah.
diagnostic. How neat.
Mhm.
What's next for you, Olivia? Where do you want to take this? Where do you see yourself in the next 5 to 10 years?
Yeah, I'm working, I think this summer I'll be writing the proposal for my dissertation, and that's going to focus on mental health and puberty, as well as body mass index in adolescence. And so hopefully be writing that up and hopefully finishing up my time here at Purdue in the next year or so. And I'd like to probably get a postdoc in a similar related field and kind of like grow my expertise a little bit more. And then I hope to apply for professor jobs if everything goes well and I'm still enjoying things. Yeah.
Awesome. So you're going to continue in the academic route and go the professor route. Excellent. Good for you. Is it okay if I ask you a couple questions about your work and about some of the stuff that you were presenting in terms of, you know, these results, the higher cortisol levels shown in the pregnant moms, I guess, reflected on the newborns, and the baby's activity, would you say hyperactivity, or how is that measured? What's the indicator there that tells you there is an effect?
Yeah, so that was, we haven't done a ton with that, but that was kind of based off the undergrad project that we did. And so, yeah, the finding was that lower hair cortisol was associated with more negative infant temperaments. And the way that that is measured, so same hair cortisol, just like I explained, but then infant temperament, excuse me, is measured by questionnaire of the mom's report of the infant. And negative kind of refers to, I think there's a couple different domains of it, but maybe being like shy or reactive to new things. I'm forgetting exactly what the questions are, but kind of like this kind of like negative affect type cluster of characteristics. Yeah, sorry, I'm forgetting like a specific example of that. That's kind of how that is measured. And so more difficult or negative infant temperaments has been associated with like poor mental health in childhood. So that's kind of one of the reasons why we're interested in that. Yeah.
And does that progress then to your interest in studying adolescents and how they're affected? Is that still connected to the pregnancy, or are you just studying their own stress levels and their own cortisol levels?
Yeah, I think it's all connected. So I didn't talk too much about this, but I also do a lot of behavior genetics type work. So we want to look at genetic influences for certain traits. So for example, like mental health is a very heritable trait. So we know that, children who are at risk for having more mental health problems, part of that is due to their genetics and part of that is due to the environment, including their prenatal environment. So a lot of my other work that I didn't talk about today is kind of looked at kind of these effects of either genetics or prenatal effects in adolescents on different outcomes, because we see that they kind of track along the life course. And so specifically talking about mental health, kids who are having a lot of mental health problems in childhood, may be more likely to struggle also during adolescence. Is a big time of change and development. And then, those maybe individuals who go on to develop like full-on disorders and stuff, and who may be in need of the most help or treatment and stuff like that. So that's kind of the idea.
And Olivia, if you were to say, if we were to serve the children, the babies, the adolescents better, what would we tell moms in order to have an impact? Do we tell them to relax? Do we give them more relaxation techniques, visualization and relaxation techniques?
Yeah, I think that's a difficult question and not necessarily answerable by our data, but yeah, I'm not sure what I would say to that. I think I generally think about these kinds of topics that an ounce of prevention is like worth a pound of treatment or something. So starting earlier is better. But at the same time, I think we'd be really interested in looking at these relationships in a study that's more high risk. So our sample wasn't very high at risk. It was mostly like a pretty upper middle class sample. So we're not quite sure like how these stressors measure up. But for I guess I say that all to say because there may be mothers who are in situations where they don't have a lot of control about the stressors in their environment, and so I think it would be a little bit invalidating to just tell them to take some deep breaths, kind of thing. So possibly, I think there's room for individual type interventions, which may be developing like a meditation practice or doing other stress relieving activities. But I also think there's room also for kind of like systemic level changes, and that could be like supporting prenatal care or other forms of healthcare or leave for pregnant moms and that sort of thing to reduce their stress level.
Yeah, totally. And no doubt that's a huge topic for how much leave do we get in North America compared to some European countries for maternity leave, and even for fathers as well as mothers. And it's a topic that definitely deserves deep inspection and discussion because of these effects that you're talking about, and because it does have an impact on the population in the generations to come. Olivia, such great work. I thank you so much for taking time to speak to us. Robin, do you have any questions for Olivia that you wanted to ask?
I was really fascinated by the process of uh, extracting that information, the hair. I have no idea all the different steps before you could actually analyze anything. I was in awe because I knew in the back of my mind that you needed to extract hair, but I thought you would be pulling it out at the root. I had no idea that you would actually cut along the base of the head. I thought you would need to get it from the root. So that was pretty interesting. What I was also fascinated is you had to wash the hair. And I thought by washing it and adding, you would be removing what's important. You know what I mean? Like, so that cortisol is embedded within the hair fiber itself. So it was just an incredible process to see that. And I really enjoyed your presentation. I learned quite a bit. I really enjoyed it very much.
I did too.
Yes, very much.
Thank you so much.
So yeah, Olivia, so just to address what Robin was saying, the cortisol is stuck deep inside the hair fiber. So by washing it a little bit at the beginning, and removing the oils and the dirt on the hair, you're not really changing the amount of cortisol that you potentially can extract by continuing on with the extraction process, right?
Yeah, that's safe to do, but I think, like I think I alluded to that, over time the hair can degrade or light can damage it. So it is a little bit sensitive, but it seems to be robust to washing it with alcohol. Yeah.
Mm-hmm.
Right. And then you also when you put it in the tube, you placed it with another liquid. Or was it the saliva?
I guess I'm not quite sure what you're referring to, but sorry, can you say a little bit more? What you mean in the tube?
After you washed it, and then it was a couple of times, then it was sent off, right, to be analyzed. Is that right? And
well, we ground, after we washed it and dried it, we ground it down to a powder, and then we put methanol in to extract the hormone.
Oh, okay, that extracted it. Okay.
Yeah.
Wow.
Yeah, so interesting. And with plants, we use liquid nitrogen, very much like how you're doing. And we put the beads in our tube and we throw everything into liquid nitrogen, let that get frozen, and then stick all of that in the vortex. And in the vortex, those beads would break apart all the plant tissue.
Oh yeah.
Yeah, I wonder how much faster you could be in doing your hair with something like that.
Oh man.
But we can talk. We can trade secrets, lab secrets, at another time. Olivia, I am mindful of your time and our viewers' time and Robin's time, and thank you so much for taking time to tell us about stress hormones and tell us how we measure them in hair, and a little bit about your journey and how you got into research and the journey that you are embarking on. We wish you all the best, and thank you again for giving us a glimpse into your journey.
Yeah, thank you so much for having me. It was my pleasure.
Absolutely. And well, thank everybody